this is going to be a joint act between myself and Sam, who's actually on the left-hand side. So maybe Sam, you can come up to the stage. Yeah. Okay, so ladies and gentlemen, I'm delighted to introduce Primol Skin Protection. It is a product, a revolutionary product that has been developed to improve hand hygiene. So briefly, I'm going to go through hand hygiene and some of the challenges that we face, go through evolution of hand hygiene through what we call our skin protection, present some brief results, and then a summary. So as this audience is very well aware, hand hygiene has been a cornerstone for infection prevention. It is one of the most effective ways of reducing transmission of pathogens and thereby reduce hospital-acquired infections. The WHO guidelines and the NHS strongly recommend the use of soap and water and hand sanitizers, especially alcohol-based hand sanitizers, which have been effective against maintaining good hand hygiene. The challenges that the system faces is that although hand hygiene practices have been in place for many, many years, there is still transmission of infection through contaminated hand, and this can lead to hospitalized infections. Although the practices have been in place for many, many years, there is still lower levels of compliance to hand hygiene, especially in stressful environments, clinical environments, and this actually leads to having contamination still present on hands, which can actually lead to less compliance with the standards that are actually in place, and these are more prevalent in non-developed countries. Although most hand sanitizers are effective immediately, they offer no residual effect, and that is the problem that we actually have. And in terms of providing protection, there is little to gain on. So we felt as an organization that there is an improvement in technology that's actually required to improve hand hygiene. So evolution of hand hygiene through Primol Skin Protection. Primol Skin Protection has been developed from technology from biointeractions. It has a Trident technology, which is an enhanced antimicrobial technology. This technology has been used for over 30 years, especially in the medical device industry. Class 3 medical devices have been coated with this type of technology. So the emergence of the Primol Skin Protection has actually come from that technology. So the formulations have been devised from biocompatible materials, and these have been functionalized to provide non-leaching biocompatible materials, forming a polymer matrix. After the application of the skin protection, there is a presence of a thin film on the surface of hands, which actually provides up to 48 hours of protection. This is actually unknown for hand sanitizers today. The technology works through a contact kill mechanism where it disrupts the surface of microorganisms through electrostatic interactions, and the technology is effective within 15 seconds of application. We have a unique property which we call kill on touch. The property, in fact, allows a surface to be cleaned whilst it's actually being touched by hands which have been treated by the hand sanitizers. So this is a very unique phenomena or a feature of the technology that's actually emerged through the use of these materials. The combination of non-leaching components and the contact kill mechanism allows us to achieve a great level of efficacy. We've actually seen 5 log reduction on many of the microorganisms that we've actually tested the product against that. We've done extensive testing on multiple microorganisms, especially at external labs, and this has included testing against viruses, bacteria, as well as yeast. We have proven that the Primol Skin Protection is actually superior to the current regularly used hand sanitizers that are actually in place today. And now I'm going to present some of the key findings from the results that we've actually obtained from these testing. And In addition to the laboratory-based results, we've actually done real-in-world use studies to demonstrate that this is actually a product that is effective in a real-world situation. So for those of you who are not microbiologists, the higher the bar, the more effective the product is. So this is a slide, in fact, looking at the virucidal activity that we've actually tested for our product. As you can see, we have achieved more than 5 log reduction, and this is excellent in terms of viruses. We've got broad spectrum activity against non-enveloped and enveloped viruses. And one of the viruses that is really difficult to kill is norovirus, which many of you are actually aware of. And this product allows us to actually kill that virus, which is not able to be killed by many of the hand sanitizers that are used today. We've also tested the product against bactericidal and yeasticidal. And then the graph on the left-hand side shows the effectiveness of the product against a number of bacterial strains. As you can see, we've again achieved a greater than 5 log activity against those strains, as well as yeast, activity against yeast. So it shows excellent activity against both gram-positive and gram-negative bacteria. On the right-hand side, we've also tested the product against drug-resistant bacteria. A number of strains have been tested, and the unique feature of this product is that it has a fast kill mechanism, and we achieved very, very good efficacy within 15 seconds. This demonstrates that the product is very effective at very short time points. We've also looked at mimicking the use of the product on humans, and we've conducted some studies using the EN standards that are required for testing of surgical hand sanitizers. On the left-hand side is a comparison that we've done with a regularly well-known used reference product and our own hand sanitizer. And as you can see, there is a great level of activity. We have a better efficiency with our product than compared to the reference product. On the right-hand side, we've actually looked at the immediate effect and also the 3-hour effect of the use of our hand sanitizer. And the graph clearly shows that we've got significant efficacy against microflora at immediate and at 3 hours. One of the unique properties of Primal Skin Protection is that it has residual activity over 48 hours. And in order to demonstrate this, we've conducted studies on pig skin as well as human volunteers. On the left-hand side is the graph showing the pig skin study. In this study, the pig skin was sprayed with our skin sanitizer, and then we basically inoculated the surface of the skin at 0, 24 hours and 48 hours time points. And then the counts were actually measured, and as you can see, there is very little change in the efficacy of the product across the 48-hour time period. On the right-hand side, we've done another test, which is on human volunteers, where the product was applied and then tested, sorry, I can't see the slide very well, where the product was tested immediately and at 48 hours. As you can see, the reference product had effect immediately, but at 48 hours it had negligible activity. So this again proves that the skin protection is providing prolonged protection over a long period of time. And this is a very, very unique feature of this product. There's another unique property of this technology, which we refer to as Kill-on Touch. And this property allows us to demonstrate that it has the ability of disinfecting surfaces that the treated hand comes into contact with. The methodology that we actually used was a combination of surface as well as hand testing that's carried out by EN standards. So in this case, the stainless steel discs were inoculated with E. coli, they were left to dry. Then the hands of the volunteers were treated with either the reference product or the skin protection product. And then after that, the stainless steel discs were touched by the hands of the volunteers, and then the counts on both the disc as well as the hands were determined. The graph on the left-hand side shows that we achieved significant effect on both the hands and on the stainless steel discs. Whereas with the reference product, there was very little activity, almost insignificant activity on both surfaces. So this unique benefit of Kill-On Touch allows treated hands to disinfect surfaces that they come into contact with. So in conclusion, we have a highly effective product which comes from biocompatible materials, giving broad spectrum of antimicrobial activity, demonstrating it has efficacy against enveloped and non-enveloped viruses, gram-positive and gram-negative bacteria, as well as yeast. The mode of action is extremely fast. We achieved this in 15 seconds, and this was demonstrated by the results that we obtained from the drug-resistant antibacterial tests that were conducted. The technology remains on the hands for 48 hours while it maintains its efficacy. This provides enhanced personal hand hygiene, helps to improve compliance to hand hygiene standards, and thereby enabling reduction of HAIs. This is the only product that allows disinfection of surfaces that it contacts, and this is a unique feature of Kill-On Touch. It leaves the surfaces much cleaner, therefore it reduces the transmission of pathogens. We have proven that the product is superior to regularly known used hand sanitizers on the market today. Please visit our booth, which is booth 22, for more information. We've done the standardized testing, but in addition to that, we've actually done a real-world use study, and that was conducted by Perfectus Biomed, and Dr. Sam Westgate is going to present that data now.
Hello everyone, so I'm Dr. Samantha Westgate, and I'm here today to present about a study that we carried out for Primal that goes above and beyond the standard methods that you need to carry out in order to demonstrate the basic level of efficacy to get a product to market. So the study was funded by Primal and carried out at Perfectus Biomed laboratories. So I'm going to run through a little bit about Perfectus and who we are as a company, then talk about hand sanitizers and what they need to do to get to market, but then also think about how you can go over and above that to see how the product works in the real world and demonstrate unique selling points and unique abilities of products that might not be demonstrated by the basic testing. I'm going to introduce the real-world in-use evaluation that we carried out at a care home in Manchester and present the high-level findings. And then this talk will be carried on at a workshop tomorrow by Dr. Kerry Ann, who's going to present more detail about the study and more detail about the results. So Perfectus Biomed is a contract research organisation. We work with medical device companies and biocide companies who are developing products against infection. So we do efficacy testing and safety testing to support that, but we're looking at a whole range of products from hand sanitizers and biocidal products through to implantable devices. But they all have something in common. They're trying to either kill microorganisms or be less susceptible to the attachment of microorganisms such that there's better patient outcomes. For this study, we were working on a product that was already on the market, but we worked with companies from early stage product development through that pathway to commercialization, and then after commercialization as well, because there's a bar to get to market, and then there, as everybody knows, in terms of products that are on the market, there's a whole range in their capabilities. So we helped pull that out as well. So in terms of getting a hand sanitizer to market, as we've just been through, there's a list of standards that you're expected to carry out. But in between 2020 and 2022, companies flooded into the market trying to make the next hand sanitizer, getting recipes off the internet, and generally that base level of data that people were producing before they put something on the market, for very obvious reasons within COVID, but it went down. And as you would expect, as we got into 2022, some of those products just disappeared again, but some people continued to support and develop their products to get further. So the very basic tests that you would do are suspension tests. You put the organism, be it virus, bacteria, fungi, into a liquid suspension. They're equally distributed within that liquid suspension, and it may or may not contain interfering substances that are slightly more representative of the real world. But in terms of the ability to kill organisms within that test, it's about as straightforward as you're going to get. It doesn't take into account a lot of the real world. So it's, I would argue it's the first thing you need to do to show that you have some efficacy in your product, not the last thing that you do. The next tests that then come in are things like the hygienic hand rub and surgical disinfection. And those methods start to use people's hands, so they're a little bit more real world, but they are very controlled, typically carried out within a controlled setting. So you get slightly more real world data, but you're not getting the variation you get in a real world clinical setting. So when Primal approached us, they wanted to take it a step further. They had a product, they could put it on the market, but they really wanted to know how it performed with real people in their everyday life going about their jobs. And so they commissioned us to set up a study in a care home. That's the first study that we've done, looking at the immediate activity of the product, but also the residual activity of the product, and also how the product behaved within the gloves. So looking at kind of fomite transfer of organisms onto a surface and the amount of organisms. So before we started the study, a lot of more traditional clinical trials use swabs to collect data. Over the years, I found that to be quite limiting in that often using a swab only collects a fraction of the organisms that are actually present. And if you don't collect enough organisms, then you can't compare products very well. The numbers are too low, the stats don't work, and it all falls apart. So before we started the study, we did a lab-based study where we looked at the same samples, so somebody's hand, and we did a traditional swab sampling method where you do a figure of 8 for 10 seconds. We also use the method from one of the standards that we just presented, where you have a broth media and the person basically puts their fingers into agar and smooshes around for about 10 seconds. And then you recover from that medium and quantify. We did contact plates, which is the picture I've got here, and we recovered from inside the glove. And we just wanted to look at what those recoveries looked like before we started the clinical study, before we used lots of people's time and money, and then found out that we recovered not much of anything. And straight away we saw that with the swab method, we were getting around 1 log of organisms back, very low amount to be able to differentiate anything statistically and reliably, and you would have needed a much longer study with much more people involved to see any difference, if at all. But from the more standard method with the broth recovery, we were getting above 5 log recovery. So straight away that looked like the method we wanted to use. A little bit more tricky to set up in the real world and a little bit odd for people. People are used to seeing swabs. They're less used to being asked to put their fingers in an agar plate with broth, but much better recovery. We also knew that because this was run over 10 days, we were going to have to collect the samples at one location and transport them to another for processing. And there's just time involved in that. So we wanted to check after overnighting in the fridge, how many of those organisms did we lose? And we found not quite a log. So we were still getting above 4 log, not getting above 5 logs. So we accept that in this study, we've lost some of the available organisms because of processing, but at least we're not down kind of below 1 log. The contact plate in the top corner there is basically the people participating took their hand, and just touched a plate, and that transfers all of the organisms that can be grown on the agar plate directly. And you can see there kind of yellow, white, large, small. You can see there's a mixed species population. So this collection method is qualitative. We're not counting anything, but it's just, as you can see, compared to the second plate that's there, after treatment, there's hardly anything on that plate. So it's a really nice visual representation of what's going on to support the quantitative data. So moving forward into the study, we didn't bother swabbing people's hands, we just left that. We were trying to be quick and not interfere with people's days, so we didn't want to do excess things that weren't going to benefit. We did do the contact plate, so we asked people to touch the agar with one hand, and with the other hand, we recovered by having the broth recovery, where they put the fingers in for 10 seconds. And then we also took samples from the inside of the glove, again with the liquid, not with swabs. So we wanted to assess Primel skin protection and also the standard of care alcohol that was used within the clinical setting. So in this case, it was a care home. We set up a table at the very front of the building, so we caught people before they went in, before they sanitized their hands, before they washed their hands, all these things that people are trained to do really, really well now. We needed to catch them before then in order to get a baseline of data. It was a care home in Manchester called Bofal, the Lectors Help, Bofal Care Home, and there were 35 participants over 10 days. So anybody who came into work came straight to us. Some of them came back every day over that 10 days. Some of them were working shifts, so they maybe came once or twice or 6 times over. But every time they came in, they were sampled. They were randomly assigned to Primel or the alcohol hand sanitizer, and if they came back the next day, they were swapped over. So if they did 10 days, they would have done 5 of each alternating. And we collected 4 data sets. So the baseline was the organisms that were there before any treatment. Then we immediately treated and collected again. So that was T0. Then the people wore gloves, went away, did their job, and came back to us in an hour. If they'd had to change their gloves in that time, they put their gloves into a packet and brought it back to us. If they were still able to wear that same set, they brought those back to us as well. So then after an hour, we sampled again, and then we took the broth and put it into the gloves, and we sampled from that too. And then this is the setup that we had to get the contact plates, the broth recovery, the internal glove, and then perform data analysis. And like I say, this is a summary. Kerry-Ann will go into more details tomorrow. But we found that, as you would expect, because we randomly assigned people, the baselines were not statistically significantly different. So we were finding, just like in the control, around 5 log of organisms on people's hands before they did anything in the morning. Then by T0, so the immediate treatment, we saw a 75% reduction in the organisms on the hands. It looks smaller on this graph because it's in log reduction, but we saw 75% of a reduction with the Primel skin protection versus a 54% reduction with the standard alcohol hand sanitizer, which is a product that's on the market and working, so it's obviously not going to do nothing, but we had a 25% improvement there, 20%. Then the T1 is when they came back after an hour, and to me this is one of the most interesting findings. So if you see how the lower line continues flat, it's because it retained that ability to reduce the level of microorganisms. So it went from 75% immediately to 72% after 1 hour, compared to the alcohol gel that was only improving the situation by 25% compared to the baseline. So an hour later, the hands of the people with the alcohol hand sanitizer were 25% better than when they did nothing at all. We didn't test into 2 hours and 3 hours and longer, but it would be interesting to see how quickly that alcohol hand sanitizer effect disappeared versus how long the effect from Primel was sustained. And then the T2 in the box at the end is the gloves. So this was quite a unique sampling method, but we put recovery broth into the gloves, mixed it around, and then quantified what was in there. And we found 85% less organisms in the Primel skin protection treated hand gloves than we did in the alcohol ones. So compared to one another, because there's no baseline for that, you got 85% less organisms with the sustained effect Primel treatment. So in summary, the Primel skin protection achieved a greater log reduction straight away. And this was from the hands of the volunteers. And then an hour later, that was sustained as well. So you saw a greater continued reduction from the hands, from the contact plates, and from inside the gloves. So in terms of future work, like I say, Primel were really keen to do an in-use study, and they want to keep collecting post-market data. They want to keep understanding how their products work in a clinical setting. So they're actually looking for 2 more settings to run the same study or a very similar study. We want to do this in order to compare the standard of care with Primel, but we also want to do it to continue to improve infection prevention and to make it a safer place for healthcare workers and to make it a safer place for the patients that we're treating. And if we continue to develop better and better products, it reduces the burden on antibiotics and other prescribed medicines because those infection risks are reduced. So the team will be at booth 2022 today. And then there's a workshop tomorrow at 11:30 that Kerry-Ann will provide more details on the study. Thank you.